Okay, so last sub-lecture of set number 2. So this I think is 2.4, 2.4. And I have very lovingly drawn out all the stuff that we need for this little discussion. And as you can tell, it's going to be on proteins, which are moderately important, you know, like just the building blocks of all life forever. Little bit important. So the very, very basics of protein is what we're going to go through today. We'll talk about amino acids, how they're made up. We mentioned DNA, and we'll go through kind of the central dogma of biology a little bit later on, but I'll give you an introduction to how proteins are formed, and we'll also go through the structure of the amino acid, which I think is very important. So, proteins, what are they? What do they do? They are linked polymers of amino acids, right? Linked polymers of amino acids. And the way that they link together is a polypeptide bond, which we will discuss in a little bit more detail. And the way that proteins work, they do a whole bunch of different stuff. And the combination of those amino acids is what gives them their structure, and their structure imparts function. So the shape of the thing determines what the thing does. In the same way that a key is different to a tennis racket, they have different shapes, and keys are good for unlocking things, and tennis rackets are good for hitting things. They have different shapes, they have different functions. In this case, we get our shape from our amino acids. Now let's look at an amino acid. So this is an amino acid. It has an amino group and a carboxyl group. So this amino group and this is a carboxyl group. It's a really good idea to know this. Okay, and then they have their kind of central carbon. Yep, central carbon in the middle. And I'd suggest it's quite a good idea to try and memorize this structure because it's just good to know. But we have our central carbon, it's connected to one hydrogen. Then we go out to our amino group, which has one nitrogen and two hydrogens, and then over to our carboxyl group, which is a carboxyl group for those of us who are familiar with chemistry. Now, what is a polypeptide bond? How do we connect these amino acids together? And indeed, what gives these amino acids their function? Well, let's answer the second question first. So this here, R, does not correspond to an element bonded to our amino acid. It corresponds to an amino acid side chain. And amino acid side chains, there are 20 of them, and they have all different functions. They're polar and non-polar, and they're charged, both basic and acidic charges. And they impart various properties to our amino acids, and therefore to our entire protein structure. They allow it to form our primary, secondary, tertiary, and quaternary structures of our proteins. They allow for hydrogen bonding and disulfide bridges. They allow for the conformational changes of the proteins in various circumstances. They make things attracted and repelled against one another. They make hydrophobia. They make hydrophilia. They cause the entire functionality of the proteins, and our amino acids tie them all together. So this is our amino acid. It has an R section which gives it functionality, and they bond together with these peptide bonds to make our amino acid branched, or our amino acid chain. They're not branched. And so let's look at the structure of those bonds. So you get one of these hydrogen off your amino group and your OH off of your carboxyl group. And in the same way that we were bonding together our glucoses, they come together in a dehydration reaction and they make a chain. Now this is a 2 amino acid chain, but the important thing to note is we have a C terminus and an N terminus. And that directionality is very important. That directionality is very important because the way in which our proteins are created is unidirectional within the ribosome. But we'll talk about ribosomes a little bit later on. So, this structure is what gives our amino acids. Remember this, I implore you to draw this structure of the amino acid. And this bond makes up our proteins, and our proteins give all of the structure and function to our cells. Well, let's not go that far. But these amino acids and their bonding and their side chains give all the structure and function to our proteins. So, let's talk about the secondary and other structures of a protein. So, the primary structure of a protein is this guy. Is the amino acid chain. It's the amino acid chain, right? Now, like I said, there are 20 amino acids. I think there are 22 if you count the non-organic ones, but 20 amino acids. And they come together in whatever order, but say the average protein is 40 amino acids long, so there's 40 to the power of 20. It's a lot. There's plenty of combinations that we can make with the amino acids that we have. So that's the primary structure. Now, our secondary structure are alpha helices and beta sheets. And these are typically stabilized by hydrogen bonds, and, you know, they're helices. That's a really terrible helix, but they're helices, and they are sheets. And the way in which we organize our amino acids and their side chains confers the structure of these alpha helices and beta sheets. Now, the tertiary structure is the combination of these alpha helices and beta sheets. So, let's take an example of a transmembrane protein. A transmembrane protein. This is not a bad example of this at all, actually. And so a transmembrane protein needs to be hydrophilic on the outside, hydrophobic where it touches the internal surface of the membrane, and it needs to be hydrophilic on the inside again. So there are a number of ways in which we can accomplish this. You can use just one alpha helix, right? So you can have an alpha helix which is hydrophobic on this surface, and then you can have a little end group which is hydrophilic, right? But the exterior surface of that amino acid alpha helix, that alpha helix, can be just hydrophilic on the outside. And then you can have your additional structure, which is a tertiary structure in it, which has this secondary kind of thing on the inside. So a combination of multiple alpha helices and beta sheets makes up our tertiary structure. Our other example that we will convert is you can have a number. This is a horrific drawing, but bear with me. To perform the same thing, and we're going to look at this from above, if you wanted to create a gap in your membrane, you could use beta sheets. But beta sheets don't allow you to curl around and have all hydrophilic surfaces or hydrophobic surfaces on the outside. In fact, they just have an outside and an inside, and you can have an outside that's hydrophilic and an inside that's hydrophobic. So how do you deal with that? You build a barrel of beta sheets, the outside of which can be hydrophobic and the inside of which can be hydrophilic. So the outside here is hydrophobic and the inside here is hydrophilic. And this structure needs to be multiple beta sheets, because you can't create that wrapped around structure with just one beta sheet. Or indeed, you might actually need to go to tertiary or quaternary structure to achieve the same thing. So that was all in one polypeptide, alpha helices and beta sheets. And then to get the next level of complexity, and you might need to do this, you could combine multiple polypeptides. Quaternary, multiple polypeptides. And that's really important because when we combine multiple polypeptides, we can increase the complexity of our quaternary protein structure. And then even it goes all the way up to macromolecular complexes, which are really, really, really big groups of these proteins, of these polypeptides. And that is a very, very, very quick introduction to proteins. Thanks, guys.